There's an important concept that I've put off talking about up until now, but it's become unavoidable at this point. We need to address the alternation of generations, which is an important feature of plants, and it's shared by all plants, but it's not a synapomorphy, because it's shared by a much larger array of multicellular eukaryotes than just plants, including most kinds of algae. Animals, which include humans of course, do not have alternation of generations, and so the concept takes a while for most students to get used to. And I'm going to use the sexual cycle of animals, which I'm going to assume that you're familiar with as a starting point. You should know, for example, that the diploid stage of a human starts off as a zygote, resulting from the fertilization of an egg which carries 23 chromosomes from mom, with a sperm from dad also carrying 23 chromosomes. This diploid, 2N, or 46 chromosome zygote, undergoes lots of cell divisions and develops into a multicellular blob, which then later becomes a baby, which then undergoes further growth to become an adult, and its development continues into geezerhood and finally death. Through all of this, one thing stays the same, and that's the number of chromosomes in each cell of the body. 46 chromosomes from zygotehood to death. The only exception to this is in those very few cells that participate in reproduction. Certain cells of the ovaries in the female undergo a meiotic cell division to produce haploid eggs, while in the males, spermatocytes undergo meiosis in the process of making sperm, which are also haploid. The next generation starts when a sperm and an egg from the previous generation fuse to make a zygote. Fertilization. Sounds familiar, right? That's basically your 5th grade sex ed class in 30 seconds or less. Well, plants too are diploid organisms that start off with the fertilization of an egg with a sperm. This diploid zygote grows and develops through a sequence of seedlinghood, young plant, maturity, and death, just like any animal. The multicellular diploid plant, called a sporophyte, also undergoes meiotic cell divisions to produce haploid reproductive cells. Only instead of gametes, the haploid products of meiosis are called spores. While animals have gonads for the production of gametes, eggs from ovaries and sperm from testes, the sporophyte produces and matures and releases the spores from structures called sporangia. You might recall from the previous video that polysporangiophytes are different from the earlier plants by having multiple sporangia produced from a single sporophytic plant. The spores, which are haploid, remember, don't undergo fertilization. And here's where the big difference from animals starts. A single haploid spore grows into the next generation of multicellular plant, which is called the gametophyte. This is the part that animals don't have in their life cycles. For us, the sperm or the egg is the haploid that never grows into a multicellular haploid, ever. It's just not the way that animals do things. Plants, on the other hand, always have this multicellular haploid generation growing out of the haploid spores that are produced by the diploid sporophytes. The gametophyte then produces haploid gametes, but without meiosis, because the gametophyte is already haploid, right? In plants, there are 2 kinds of gametes. Eggs are differentiated in structures known as archegonia, and sperm are produced in the antheridia. For a plant like a moss, having the archegonia and antheridia close to the ground is best, because the sperm have to travel through the water in order to get to the eggs. Moss antheridia release the sperm on a rainy day, and the sperm travel in the floodwater to an archegonium that encases the egg that the sperm must fertilize. Once the sperm is fused with the egg, fertilization, the resulting zygote then grows into a sporophyte, which is where the cycle starts over. There are 2 generations here, the diploid sporophyte and the haploid gametophyte. Technically, the gametophyte is always the offspring of a sporophytic plant, while the gametophyte must achieve fertilization in order to have reproductive success. Its offspring must be diploid or sporophytic, hence the idea of alternation of generations. There's one more idea that I want to tack on here, and it's also effectively a pretty good synapomorphy for the plant kingdom. It has to do with how the sporophyte growing from the fertilized egg is always still attached to the gametophyte at whose archegonium the egg was produced. Let's say on this gametophyte right here, we've got an archegonium, and inside the archegonium we've got the egg. After fertilization, the baby diploid plant grows and it gets nutritive assistance from its mom, the gametophyte, for at least the first part of its growth. This is what it means to be an embryophyte. The embryophyta or the embryopsida are both terms that are used more or less interchangeably with the plant kingdom. Kingdom Plantae is about the equivalent to the embryophyta. In hornworts as well as mosses and liverworts, the sporophyte completes its entire growth while attached to the mother gametophyte. It's like having that child that grows up, gets married, and never ever leaves the house. You probably know somebody like this. You never see a hornwort sporophyte that's not attached to its gametophytic mother. Polysporangiophytes, though, like Cooksonia, differ not only in having multiple sporangia. The sporophyte generation is more extensive. It sprawls around and outlives the gametophyte that it initially grew from. These sporophytes can take advantage of their upright stature and further growth, and they bear their sporangia at the tips of vertical stalks, and this gives them an advantage when it comes to dispersing their spores, which have to be carried by the wind. Remember the 6.5 centimeter height attained by Cooksonia? That was probably towering in comparison with the other plants of the time that were more flimsy. Here is a more fleshed out tree of life, showing the relationships among the plants that we talk about in this lesson. At the base of the tree over on the left hand side, this tree is the same as the one I had up in the previous video. Now in the previous video I described Cooksonia as vascular. Technically this isn't true. While Cooksonia did show a very primitive vascular-like protostele structure that was adequately reinforced to allow this growth to 6.5 centimeters and also to make these plants somewhat fossilizable. They didn't have dedicated separate structures for the movement of water, which would be xylem, and sugars, which would be phloem. These specialized kinds of vascular cells don't appear until later. In the tree, Cooksonia, as well as a couple of other plants, branch off the tree as polysporangiophytes that are not part of the true vascular plants or the Eutracheophyta. At this point you might ask, how can we really tell what kinds of cellular structures were there in these Silurian and Devonian plants? Are the fossils that good that we can put them under the microscope and identify different cellular structures? Well, certainly some fossils are really nothing more than vague smudges on a rock in the shape of a leaf or a branch. At best, a two-dimensional projection of what was originally a three-dimensional structure. And to be sure, you won't be able to get very good microscopic pictures from this kind of fossil. And then we have the Rhynie Chert, which is a fossil site in Scotland. Chert is a type of rock that forms in hot springs habitats. And what happened at Rhynie is that the late Silurian and early Devonian plants, animals, and fungi that died in this mineral pond, they got encased in silica-rich materials, preserving their 3D shape. Their cell walls were actually fixed in place by this mineralization, so that by making thin sections of this rock, the chert, and observing them under a microscope, you can see the same level of detail that you would in modern preparations made with modern plants. Paleo Porn Alert. Here are some images from the Rhynie Chert, showing the protostele-like structures in cross-section from the stems. Aglaophyton, Horneophyton, and Rhynia are three kinds of polysporangiophytes that were taller and larger plants than Cooksonia. Like Cooksonia, they were not officially vascular plants, although they did certainly have tubular structures that permitted them to grow and thrive in this ancient Rhynie hot springs habitat. Here's where I put the Rhynie Chert plants in the tree. Now the Eutracheophytes are the truly vascular plants with dedicated xylem and phloem, and although we don't have them in the Rhynie Chert of the Devonian, they actually first appear in Silurian fossils before the Devonian. Baragwanathia is the earliest Eutracheophyte in the fossil record, and it comes from the Silurian. Now the structural advantages of true vascularity gave these plants the potential to achieve much greater size, and by the end of the Devonian, these are the plants that had evolved into the tree-sized oddities that eventually took over the continents during the Carboniferous. By the way, this is where most textbooks gloss over the distinction between the Polysporangiophyta and the Eutracheophyta. If you don't worry about plant lineages like Cooksonia or the plants of the Rhynie Chert, You can just say that all the plants, except for the non-vascular liverworts, mosses, and hornworts, are all vascular plants. But in the interest of developing a clearer picture of the grades of vascularity that we do see building up over the course of early plant evolution, I wanted to be really inclusive here, so I decided to keep these two things separate. The picture that we get from the fossils here is one of a carpet of green that gets taller and taller over the course of the Silurian and Devonian. The more the plants grow, the more soil there is underneath the plants for the plants to use, and these plants are under continual selective pressure to become taller and more robust. The eutracheophytes happen to win there because of the way that their vascular tissue permitted larger size. The two eutracheophyte lineages are the Lycophytina and the Euphilophytina. Lycophytes are still around today in a pretty minor presence, but they are very important players in the Carboniferous forests. The biggest group of modern lycophytes are the club mosses, which look sort of like clubby-shaped mossy plants. And then you've got the spike mosses, represented famously by Selaginella, sometimes called the resurrection plant because of the way it tolerates being totally dried out. It's like the perfect plant for somebody who forgets to water for long periods of time, because even after it gets totally dried out, it will spring back to life if you add the water. There's a favorite demonstration that every botany student gets to see, which is where the professor drops out this dried out ball of twigs into a bowl of water. And then by the end of the lecture, this Selaginella plant looks like a happy green plant again. Much larger lycophytes, tree-sized club mosses like this Lepidodendron fossil. They were prominent in the Paleozoic coal forests. Lepidodendron in particular comprised up to 2/3 of the biomass in wet equatorial lowland forests. Long-term burial of their biomass is what resulted in the massive coal deposits found across North America, Greenland, and Russia. Now this was basically the ideally adapted swamp tree, living in some of the wettest, swampiest conditions the planet has ever known. So let's talk a bit about atmospheric carbon. By the time we get to the Carboniferous, tens of millions of years of photosynthesis by plants on dry land would have pulled away lots of carbon dioxide from the atmosphere and stored it in standing forests, and under the swampy conditions like those preferred by Lepidodendron, large amounts of that carbon got buried to form coal. Now there's another non-biological dynamic in which the atmospheric CO2 is removed by the weathering of silicaceous rock, and this happens to be sped up by the activity of plants. Over a long course of time, starting mid-Devonian and continuing right through the Carboniferous, the removal of CO2 from the atmosphere resulted in a reverse greenhouse effect, where the thermal blanketing of atmospheric carbon was drastically reduced, and this would have had a profound cooling effect on global climate. This is more than just idle speculation. It's supported by those Earth system models we talked about before, and also geochemical proxies like carbon-13 excursions. We also have biological proxies for evaluating climatic or atmospheric chemistry trends, and this is what I want to introduce here. Remember stomata? They're the tiny holes on the stems and leaf surfaces of plants, through which the plants take up carbon dioxide to use for photosynthesis. Well, it turns out the density of stomata, the number of holes per square millimeter on leaf surfaces, it changes over time according to what's needed by plants for a given level of atmospheric CO2. During periods of high carbon dioxide, the plants are able to meet their CO2 uptake needs with relatively few stomata. But when the CO2 levels drop, the plants require a lot more stomata in order to get an adequate uptake of CO2, which is now a lot rarer. Atmospheric carbon levels and plant stomatal densities both go up and down over the course of the history of land plants, and they do so in an inverse relationship. When CO2 levels decrease, the fossilized leaves of those periods show an increase in stomatal density, and when the CO2 levels increase, the plants respond by making leaves with fewer stomata. This is an example of a biological proxy. We're using a feature of the organisms living in the habitat as an indicator of the atmospheric CO2 levels. So back to our story. The lycophytes, Lycophytina, they had their day in the sun during a period in the Earth's history when things were extremely wet. But as things dried out, both with the cooling of the global climate, remember less evaporation off the oceans, as well as the formation of the supercontinent Pangaea, the other kinds of plants came to dominate, and the lycophytes sort of retreated back into minor ecological roles. The other group of eutracheophytes is the Euphylophytina, which is the main group that contains nearly all modern plants, ferns, gymnosperms, and angiosperms. They're all in the Euphylophytina. Again, we're going to split this group in half, identifying two subgroups. And the first of these is the Monilophyta, which is actually a pretty important group because it contains the ferns. Now, ferns are, in terms of species diversity, the second most successful group of plants. Okay, it's a very, very, very distant second place, with 11,000 species of ferns as compared to 257,000 species of flowering plants. But this still puts ferns well ahead of conifers and mosses, which are in third and fourth place. Now, besides the ferns, the Monilophytes include some other relatively common plants like horsetails and whisk ferns. Whisk ferns are not really ferns at all, even though it sounds like they have a ferny name. One thing you probably already know about ferns is that you usually find them in places where it's wet. Maybe at the margin of a lake or a stream, or near waterfalls or under the forest canopy where it's usually cool and damp. This is because of the way that ferns reproduce. The plant that you recognize as a fern is the sporophyte. Now, in most fern sporophytes, you can turn a frond upside down to see rust-colored spots or stripes. These are called the sori, singular sorus, and this is where the fern produces and releases its spores. The spores grow into the gametophyte generation, which in ferns is flimsy, soft, and tiny, kind of like a small fleck of algae, and it requires a fairly long period of very damp conditions in order to complete their growth to maturity. I think of ferns as being the plant equivalent of amphibians, like frogs. They can live on dry land, but you're always going to find them in wet areas because they have to go back to the water in order to reproduce. Modern ferns are restricted to grow only in those habitats that are wet enough to support their gametophytic generation. Now, the same thing was true with the Monilophytes of the Paleozoic. Those conditions we talked about before, the equatorial ever-wet forests of giant lycophytes, also contain a great number of giant ferns and giant horsetails. Now, some of those horsetails reached heights of 35 meters, that's over 100 feet. Some of the larger ferns grew to 10 meters, much, much bigger than what we call giant ferns today. But as this super wet habitat went away with the climatic changes caused by plants themselves and also by the formation of the supercontinent Pangaea, so did these plants. Lycophytes, ferns, and horsetails that we have today are sort of like pale, pathetic, miniature versions of those plants that dominated the lowland equatorial forests of the Paleozoic. So when I put this tree together, I was originally going to talk about a couple of other plants, but I'm going to wait until a little bit later for that. Right now, we've gotten to the point where we've talked about the Euphylophytina and this lineage over here, the Monilophyta, horsetails and ferns, and I didn't draw this thing in over here, which I could have drawn in like this and said this was going to be the whisk ferns. Yeah, those are the ferny named things that are not really ferns at all. In fact, here's a picture of one. Okay? We're going to be spending a lot more time from here on talking about the Spermatophyta, this group over here, the seed plants. And I just want to take this time to review what we did with this video. First, we talked about the alternation of generations, in which we have this sequence of events: growth of the sporophyte, production of the spores, growth of the gametophytes, production of the gametes, fertilization to complete the cycle. This concept of alternation of generations is something that I really do want you to know. Okay? It's indispensable. We can't talk about plants unless you actually have a good idea of how plants have two generations embedded in their life histories. Okay? The second thing we did was to review our tree and actually to develop the tree. I gave you the old tree, the tree from the previous lecture in green, and I used this color of congealed blood, kind of a purplish brown, to talk about the Eutracheophyta, really the truly vascular plants, which included the Lycophytina, the club mosses, and the spike mosses, as well as a really important tree from the Carboniferous and Permian forests, Lepidodendron, and the Euphylophytina, which includes two groups, one of which, the Monilophyta, ferns and horsetails, we talked about them. kind of in the same line as Lepidodendron as being some of the most important plants of the Carboniferous forests, kind of tapering out and fading back as the Permian got drier. And eventually, as we move into the Mesozoic, thing is going to get more and more dominated by things like gymnosperms, which are spermatophytes. And that's going to be the group that we tackle next.